Welcome to SL Biologica, a foundation to life science. In this video, we are going to discuss about amino acids and protein structures. So what are proteins? We know that proteins are important biomolecule in our living system, isn't it? These proteins are made up of amino acids. Consider this protein as a polymer form, then its monomer or basic structural unit form is amino acid. A set of 20 amino acids makes up proteins. So this is a basic structural unit of an amino acid. Then we can say central carbon atom, alpha carbon atom. It is attached by an amino group, carboxyl group, hydrogen atom, variable side chain. Okay. So based on this variable side chain, amino acid differs. So an amino acid is made up of an amino group, a carboxylic acid group, a hydrogen atom, and a characteristic R group connected to a central carbon atom known as alpha carbon. So the side chain is another name for the R group. Alpha amino acids are chiral because they are attached by 4 different groups. In previous slide, we have seen that it is attached by amino group, carboxyl group, hydrogen atom, and one side chain or R group. These are applicable except in glycine because glycine is the most simplest form of amino acid. It consists of hydrogen in its R group. Okay, they can exist in either of two mirror image forms known as L isomer or D isomer. So basically, L isomer is the most exist form of proteins. Levorotatory isomers and destrorotatory isomers. Okay. So these are the representation of 20 amino acids. Based on its side chain, amino acids are divided into different classes like polar, non-polar, charged, uncharged, negatively charged, positively charged, aromatic amino acids, etc. So we already discussed about its basic structure form. These amino acids are connected by a bond known as peptide bond. Okay, it has two terminus like C terminus and N terminus. Okay, and then this is a non-polar uncharged group. So we already discussed that based on R group, amino acid differs. So glycine is the most simplest form of amino acid. And then there comes cysteine, proline. So this all amino acid has different type of R group. Okay. So non-polar uncharged classes include alanine, valine, leucine, isoleucine, methionine, glycine, cysteine, proline, etc. So these amino acids are represented by 3-letter code and it is also indicated by one capital letter. So glycine here you can see GLY or capital G. Here we can see aspartate as ASP and it is also represented by one capital letter, capital D. So here we have to note about this capital letters. So these are the aromatic amino acid. We can say aromatic side chain. Here, tryptophan, phenylalanine, tyrosine. Then these are the negatively charged amino acids, aspartate and glutamate. And it is positively charged amino acid, lysine, arginine, histidine. These are basic amino acid. These are acidic amino acid. Aspartate and glutamate are acidic amino acid. Then there comes polar uncharged. Polar meaning it will contain OH groups in amino or amide group in its side chain. So examples are serine, threonine, asparagine, glutamine. Here, check this one. Asparagine is represented by capital N, glutamine is represented by capital Q. Okay. Each amino acid has its own properties and its own characteristics. Here we will discuss about one of two important amino acid. This one is proline. In contrast to other members of the set of 20, proline has an aliphatic side chain that is connected to both nitrogen and alpha carbon atoms. Okay, so this is it has a aliphatic side chain uh, that is connected to this nitrogen and alpha carbon atom. So proline has a different conformation, right? So it acts as a alpha helix structure breaking form. Okay. Because of its ring shape, which makes it more conformationally constrained than the other amino acids, proline has a significant impact on protein architecture. Okay, so because of this structural characteristic, uh, it play an important role in protein architecture formation. Okay, then we will discuss about the next amino acid, histidine. Histidine has a imidazole side chain. Okay, an imidazole group, an aromatic ring that can also be positively charged, is present in histidine. Okay, this histidine imidazole side chain has a pKa value that is related to pH that is close to 6, or it can be neutral. Mm? So its imidazole side chain pKa value is closely related to 6 or 7. Okay, it can be either positively or uncharged near neutral pH. So likewise, each amino acid has its own properties. So we have discussed about two important amino acids, proline and histidine. Then this amino acid in neutral pH solution primarily exists as dipolar ions. It is known as sweeter ion. It have both positive and negative charges. Okay, this dipolar form, the amino group carries a positive charge amino NH3 plus, while the carboxyl group has a negative charge carboxyl COO minus. The ionization state of an amino acid changes depending on the pH level. Okay, so the amino acid will be neutral at this pH. So that form of structure is known as sweeter ion, where we can found both charged group, like negatively charged carboxyl group is present there, and positively charged amino group is also present there. Then let's discuss about peptide bond. Peptide bonds are essentially planar in nature. Okay, so these amino acids are connected by peptide bond. This peptide bond is planar in nature. They have a significant double bond nature, which limits the conformation of the peptide backbone by preventing rotation about this bond. And it is represented by two dihedral angles. Dihedral angle means angle presenting in the half plane of a molecule. Okay, here we can see NC alpha bond is called phi, NC phi, like that you can remember. And CC alpha bond is called psi. Okay. Then this conformation of protein is represented in a diagram known as Ramachandran diagram. The allowed values can be visualized on a two-dimensional plot called a Ramachandran diagram. It was discovered by an Indian scientist called Ramachandran. Here we can see 4 quarters, in which 3 quarters we can see the secondary structural forms of protein. Here it represents the allowed conformation of a protein. X-axis represents phi and y-axis represents psi. Phi is the NC alpha bond. Psi is the CC alpha bond, right? So 3 quarters of the possible combinations are excluded simply by local steric clashes. So based on its conformation, for each plot we have representing each conformations of protein, like this first quadrant is represented by alpha helix form. right-handed alpha helix form, and second quarter is represented by beta plated sheet form, and third quarter is represented by left alpha helix form. Okay, steric exclusion, the fact that two atoms cannot be in the same place at the same time, can be a powerful organizing principle. So based on the steric repulsion and steric exclusion, the scientists represented this conformations of protein in this diagram, right? So not all psi and phi values are possible without collisions between atoms. Okay. Then coming to the structure forms of protein, first comes primary structure of protein. This is the basic structure form of a protein where you can see, basic structures or basic unit of amino acids connected by peptide bond. So amino acids are made up of nitrogen, carbon, and oxygen. 20 common amino acids are used to build proteins. Here, amino acids are connected with peptide bonds, and chains of amino acids are polypeptides. So this is the basic structure forms of protein, where this primary structure of protein combined together to form secondary structure of protein. Examples for secondary structure of proteins are alpha helix and beta plated sheet. Okay. And there also exist turns and loops in the secondary structure forms. The secondary structure forms are discovered by Linus Pauling and Robert Corey. So coming to some terms like pitch of the alpha helix. What do you mean by pitch of the alpha helix? It is actually the product of translation distance and number of residues per turn. That is 1.5 angstrom into 3.6, which results into 5.4 angstrom. Angstrom meaning 10 raised to minus 10 meter. Okay. So algorithm for hydrogen bonds in an alpha helix, the CO group of first residue, that is I, creates a hydrogen bond with NH group of fourth residue, that is I plus 4 in that helix. Okay, so I is represented as a first residue. So this is the alpha helix hydrogen bonding. Okay, the first residue CO group will be bonded with the fourth residues NH group. So hydrogen bonds between polypeptide strands stabilize this beta sheet. This alpha helix is a single strand structure forms, while beta sheet, it exists as a sheet-like structure form. Okay. Here we can see intra and inter hydrogen bonding. In alpha helix, we can see intra-chain hydrogen bonding. Okay. So this is the primary structure of a protein and secondary structure. Then there comes tertiary and quaternary. Tertiary is more advanced or more complicated forms of secondary structure. That is tertiary. Then there comes quaternary. Again, the complicated forms of tertiary structure, that is quaternary. Then tertiary. Tertiary is like 3D structure forms of protein. The pattern of disulfide bonds and the spatial arrangement of amino acid residues that are widely apart in the sequence are referred to as tertiary structure. Basically, spatial arrangement or 3D structure of forms of protein is called tertiary structure. Then there comes fourth level structural organization, that is quaternary structure. This quaternary structure, each polypeptide is exists in the form of subunit. Okay, the spatial arrangement of subunits and the character of their interactions are referred to as quaternary structure. Okay, here you can see a dimer. The dimer is the most basic type of quaternary structure. It is made up of two identical subunits. So quaternary structure is made up of subunits. A dimer is an example of a quaternary structure. Okay, this all are about this amino acid and protein structure forms. Okay, thank you, friends. Thank you for listening. You can check your understanding of amino acids and protein concepts with the video given in the description, where I have discussed some important questions on proteins. Okay, friends, thank you.